Okay, so this is how so I have 3 stages of how blood becomes urine. Okay, so how we go from plasma to urine. So this is what's happening in the nephron. Okay, so these are the things that are happening in the nephron to make urine. Okay, so if we look at this, let's go over the parts first. Okay, so here's the glomerulus. Okay, so here's the renal corpuscle is this whole thing, right? And the glomerulus is the can you hear the dog panting? The glomerulus is the capillary in there. Okay, then we have this space, and then we have the capsule that surrounds it. So there's the capsule that surrounds it. Okay, afferent arteriole enters, efferent exits. Okay, so A comes before E, so that's how you're going to get in there. Here's the PCT. I'm trying not to mess it up too much. Here's the nephron. Let me get a pointer. I'm going to use the lightsaber, which will be totally dorky. Okay, so here's the PCT. Okay, here's the nephron loop, right? So the descending limb comes first, and then the ascending limb, and then the DCT, and then the collecting duct. Okay, now I want you to see up here, this is representing the peritubular capillaries. So those capillaries are actually, these are going to be wrapped around the tubes, okay, so wrapped around the nephron, so peritubular, they're around it. Okay, and then down here, this is the vasa recta, okay, so those are the ones that are wrapped around the nephron loop. Okay, now, 3 steps, and they're summarized in these boxes, okay, and the arrows are the same color as the box. So first step, glomerular filtration. Okay, so what's going to happen is the glomerulus is going to function as a filter. Okay, so basically the blood's going to come into here. The blood pressure is what's going to drive the fluid out, okay, out of here. So it's going to go into the ducts. Okay, so the movement of substances from the blood within the glomerulus into the capsular space. Okay, so this is where we're filtering. Okay, now, we are really, really good at getting things through there. Okay, so we're like too efficient at filtering. Okay, so everything gets filtered. We just pick up too much. So what we have to do is we have to reabsorb. We don't want everything that goes through there to wind up in our urine, or we're going to lose all these molecules that we've been working hard to make, and we'll lose too much water. We'll be dehydrated. Okay, so we're going to reabsorb. Okay, so tubular reabsorption means things are going to come back from the tubes and go into the blood vessels. Okay, then when we get to at the same time, other things going on is more secretion. Okay, so there are still some like toxins or wastes in the blood that need to go into the DCT and the PCT and the nephron loop. Okay, so secretion means we're coming from the blood and we're going back into the urine. So I think that makes sense. Okay, so here's another image that's from an old book, and I just left it in here because it kind of shows exactly this. It shows the same thing. Okay, so we have filtration, then we have reabsorption. Okay, then we have secretion, and then we have, this adds another step of water conservation at the very end, how we pick water back up into our blood so we don't get dehydrated. It just shows the same steps. Okay, now, what does the glomerulus actually look like if you looked at it molecularly? Okay, so the glomerulus looks, here's the capillaries. Okay, so here's the capillaries coming in. Okay, and on top of them are these cells, okay, the capsule. And sometimes we describe it kind of like a glove. Like if you put your fingers together, it kind of goes like this. Like it overlaps. Okay, so this is the filtration membrane. So it's important that this membrane is functioning properly so that the right things are filtered out and are ending up in the urine. Okay, because you don't want to lose things that you need, and you don't want to like have blood and protein go into the urine. Okay, so we have to go over the anatomy of the glomerulus and the filtration membrane. Okay, so here, I think I have it on the next slide. I have it written out too. So there are, they call these little fingers coming out of here, they call them pedicels. So they're like little feet coming out from, or fingers coming out from the cell. So the capillary has pores. Okay, so there's fenestrations. Okay, so big things are going to go through there fast. There's an endothelium, and then there's slits. Okay, so slits are going to let bigger things in. So they took this and they magnified it so you can try to see it better. I don't know if this picture explains it very well or not. Okay, so here's what we have. Okay, so the membrane is or these layers here. Okay, so here's the capillary. Okay, so that's the capillary, and then this is the glomerular capsule that's over top of it. Okay, so the capillary has holes. Okay, so it's got fenestrations in it. Okay, plus we're going to have things moving in between the cells. Okay, then we have an endothelium, right? So there's the endothelium with the holes. Okay, here's the basement membrane of the glomerulus, and then we have slits. Okay, so basically, here's what's going on, is the endothelium is blocking formed elements. So it's blocking red blood cells, white blood cells, platelets. Okay, so it's keeping them in the blood. So see, if you look here, they're staying in the blood. So white blood cells, platelets, red blood cells. Okay, now the basement membrane blocks big proteins. Okay, so here's some large proteins. So things like albumin stay in the blood. Okay, and then the slits are blocking these small proteins. So these are staying in the blood. This is what's important, is that all of this stuff is what's staying in the blood. I think that makes sense. Okay, so that should be in your blood. It shouldn't be going anywhere else. Now, if you look at this is what's ending up. So this is the capsular space. So this is what's ending up in the filtrate. Okay, so it's got water, glucose, amino acids, ions, urea, some little hormones, vitamins that are water soluble like B and C, okay, ketones, and some really small proteins, but not much protein. Okay, that's ending up in the filtrate. So this side of this sums up what's going on. And I kind of made a mess out of it. I'm not so worried that you know all the anatomy of this, but this is what's happening. Okay, so here's it written out. So what stays in the blood? So proteins, platelets, blood cells. Okay, some things that are bound to big proteins stay in the blood. So things like calcium, iron, thyroid hormone should stay in the blood. Things that end up in the filtrate, okay, water, glucose, B and C, amino acids, some little proteins. Okay, here's the waste: urea, uric acid, creatine, ions, and ketones. Okay, now, if something is wrong with that membrane, okay, if that glomerular membrane is damaged, okay, things will go through that aren't supposed to go through. Okay, so maybe if the blood pressure is too high, or someone has a kidney disease, okay, they can end up with proteinuria or hematuria, which, look, protein in your urine or blood in your urine. And you'll do those tests in the lab. We have a urine lab, and you just stick a dipstick in. Okay, so these things are going to suggest different disorders. Okay, you should know this. Okay, now, if you look at this, we end up filtering out things that we want to get back. Okay, so we're going to end up reabsorbing things like glucose, amino acids, some small proteins, water for sure. Okay, so some things we're going to reabsorb, we have to pick back up. Okay, now, what causes this to occur? The filtration occurs because of differences in the pressures. Okay, so the afferent arteriole is bigger. Okay, the diameter of it is bigger than the efferent. Okay, so essentially what that does is that guarantees that more blood is coming in than is exiting out. So it maintains a higher blood pressure in there. Okay, the hydrostatic or blood pressure inside of here. So I'm just going to put BP so you can see what it's trying to say. So glomerular hydrostatic pressure. So that's the blood pressure in the glomerulus. It's usually about 60. We try to maintain it around 60 millimeters of mercury. Okay, now there is some blood colloidal pressure. Okay, so the blood has some osmotic pressure that's going to pull fluid back in. Okay, it's going to hang on to fluid. And then there's a little bit of pressure in the capsule as well. So what happens is we end up, though, with a net filtration pressure that is going to allow us to have make urine. Okay. Now there are a whole bunch of ways that our body has to ensure that we're going to make urine no matter what's going on. Okay, so our body is really good at keeping this going. So we have auto-regulation systems to make sure that the net filtration pressure keeps filtering, okay, keeps being out. So we can change the size of the afferent arteriole. We can do a couple different things, but I hope that makes sense.